Hi, in this video we will talk about 5 fruit fly species that we use in our research. We will also briefly talk about the research context that they are studied in our lab.
We need to maintain the flies in our laboratory setting. Rearing some species is difficult, so we have to be careful of some conditions. We keep the temperature and humidity constant. In our laboratory, 2 kinds of foods, corn food and malt food, are made. We also have vials of different sizes. All flies are kept in the fly room at 25 degrees Celsius.
Drosophila melanogaster is the most famous fruit fly because this species is a model organism in various research fields. Also, Drosophila melanogaster's genome has been first sequenced in Drosophila, and therefore gene functions are relatively better known. Since its genetic structure is studied well, it's commonly used as a reference for the studies of other Drosophila species. There are a lot of mutants, and a large number of genetic lines are available.
Drosophila suzukii is also known as a pest because it can lay eggs on fresh fruit, while Drosophila melanogaster lays eggs on rotten fruit. Drosophila salpicula, a close relative of Drosophila suzukii, can also lay eggs on fresh fruits, but Drosophila suzukii is able to lay eggs on harder fruits. Also, the males of this species have characteristic spotted wings. In our laboratory, Drosophila suzukii has been compared with Drosophila salpicula to study the differences in preference for oviposition site and the evolution of genitalia morphology due to differences in oviposition site.
Drosophila albomicans is bigger than Drosophila melanogaster, and distinctive stripes are present on both sides of its legs. On males' heads, there is a white shiny structure. This is called frons. Drosophila albomicans originally lived in Southeast Asia. The former professor at TMU, Dr. Kitagawa, clarified the habitats of Drosophila albomicans in the 90s. However, their habitat zone has expanded northward since the 1990s, so this species is used in the experiment of adaptation of cold regions.
Drosophila guttifera lives in North America. This fly has a strong tolerance to alpha-amanitin, a toxic compound included in mushrooms. This fly has polka-dotted pigmentation patterns on wings and on the abdomen. A striped pattern can be seen on the dorsal side of thorax. The genome of this species has also been sequenced and it is available. So, this fly is a model to study developmental mechanisms for formation of pigmentation patterns.
In summary, Drosophila melanogaster is the extensively studied model organism in various fields, including evolution. Drosophila suzukii and Drosophila subpulchrella are closely related to each other and used in speciation studies. Drosophila albomicans seems to get cold tolerance during its expansion to the northern regions, and we are interested in the mechanisms involved in the cold adaptation process. Drosophila guttifera has a distinguishing pigmentation pattern and is used for evolution of morphology. Thanks for watching.